My dear students, today we can study, observe the illustration and answer the questions. In the first part of this video, we have discussed 16 questions. That video's link is given in the description box. Today, we can discuss the other questions. Question number 17. A. Identify the pathogen. The pathogen of AIDS is HIV, human immunodeficiency virus. Question 17b. How does the pathogen multiply in the human body? Answer. HIV enters the body and multiplies using the genetic mechanism of lymphocytes. The pathogen multiplies using the genetic mechanism of lymphocytes. Question 17c. Condition that makes AIDS fatal. Answer. HIV enters the body and multiplies using the genetic mechanism of lymphocytes. The number of lymphocytes decreases. It reduces the immunity of the body. Various other pathogens enter the body, and this makes the condition of AIDS more fatal. Question number 18. A. Identify the disease. Answer. The disease is diphtheria. 18b. Name of the pathogen. Answer. Corynebacterium diphtheriae is the pathogen. 18c. Symptoms of the disease. Toxins produced by the bacteria cause fever, throat pain, and inflammation in the lymph glands of the throat. Cells in the mucous membrane produce an ash-colored thick coating in the throat. Brain, heart, and kidney are gradually affected. Question number 18d. Organs or body parts affected. Answer. Diphtheria affects the mucous membrane of the nose, lymph glands, and the throat. The main organs like brain, heart, and kidney are gradually affected. Question 18e. Vaccination is the best preventive method. Why? Prevention is better than cure. Antitoxins act against the toxins are used to protect the uninfected cells. If the disease becomes severe, the patient cannot be recovered through medication. So, vaccination is the best preventive method. Question number 19. A. Identify the pathogen. Answer. Leptospira. 19b. Name the disease. Answer. Rat fever or leptospirosis. Question 19c. Name the reproductive method by which this pathogen multiply. Answer. The pathogen multiply through binary fission. Question 19d. The major symptoms of this disease. Answer. Severe fever, headache, muscle pain, and redness in the eyes. Question 19E. What are the measures to be taken to prevent the transmission of rat fever? To control diseases like rat fever, eliminate the vectors. Take preventive medicines. As per the direction of experts, ensure personal hygiene and environmental hygiene. Proper disposal of the waste. The realization that immunization is better than treatment is very important to stop the spread of the disease. Question number 20A. Name the diseases which are transmitted like this. Diphtheria and tuberculosis are the diseases which spread through cough and sneezing. Question 20B. Name the pathogens. Diphtheria. The pathogen is Corynebacterium diphtheriae. Tuberculosis. The pathogen is Mycobacterium tuberculosis. In both, the pathogen is bacteria. Question 20C. The best preventive method for this type of diseases. Answer: vaccination. Vaccination is the best preventive method for this type of diseases. Diphtheria, the vaccine is pentavalent or DPT vaccine. And tuberculosis, the preventive vaccine is BCG. Question number 21A. Identify the figure. Answer: lymphatic system. Question 21B. Write the function. Answer: the lymphatic system help in defense mechanisms. The lymph contains plenty of lymphocytes. They destroy the disease-causing bacteria in lymph nodes and spleen. Question 21C. Find out the major parts. Answer: lymph duct, spleen, lymph nodes, and the lymph. Question number 22A. What are vaccines? Answer: vaccines are the substances used for artificial immunization. Question 22B. Components of vaccines. Answer: any one of the components from alive or dead or neutralized germs, neutralized toxins, or cellular parts of the pathogens. 22C. Significance of vaccination. Vaccines act as antigens. It stimulates the defense mechanism of the body. Antibodies are formed in our body against the antigens. Antibodies retain in the body, which in future protect our body from the pathogen responsible for same disease. Question number 23A. Identify the picture. Answer: these are the DNA samples collected for DNA testing or DNA Fingerprinting. Question 23b. Name the scientist who paved the way for this. Answer: Alec Jeffrey. Question 23c. Basis of this technology. Answer: just like the difference in the fingerprint of each person, the arrangement of nucleotides in each person also differs. This is the basis of DNA testing. Question 23d. Scope of DNA fingerprinting. Answer: to find out the hereditary characteristics, to identify real parents in the case of parental dispute, to identify persons found after long periods of missing due to natural calamities or war, to find out the real culprit in murder or robbery. The real culprit can be identified from among the suspected person through this method. Question 23e. Things used for this technology. Answer: DNA of the skin, hair, nail, blood and other body fluids of the suspected persons. Question number 23f. How are persons identified through DNA testing? By testing the arrangement of nucleotides of the suspected one, the real person can be identified. Question number 24. Lost child found after years, identified through DNA testing. Did you notice the heading of the newspaper report? How are persons identified through DNA testing? Answer: just like our fingerprints of each person differs, the arrangement of nucleotides in each person also differs. The arrangement of nucleotides among close relatives has many similarities. So DNA fingerprinting is helpful to identify the persons found after long periods of missing. Question number 25a. Identify the picture. Answer: it is the logo of Human Genome Project. Question 25b. Relevance of Human Genome Project. Answer: Human Genome Project was started to identify the exact gene responsible for a specific trait and its location. Question 25c. Benefit of gene mapping. Answer: gene mapping helped to identify the location of a gene in the DNA responsible for a particular trait. Question 26a. What is genome? Answer: the complete genetic material present in an organism is called its genome. Question 26b. What are junk genes? Answer: in human DNA, the majority of genes except the genes that code for protein are non-functional. They are called junk genes. Question 26C. Genes in human genome are identical to those in bacteria. Answer. About 200 genes in human genome are identical to those in bacteria. Question 27A. Identify the picture. Answer. Production of insulin through genetic engineering. Question 27B. Write the different steps of this process. Answer. Human cell, human DNA. Bacteria, circular DNA of the bacterium called plasmid. Cutting of insulin gene. On the other side, isolation of plasmid. Joining insulin gene with plasmid. Plasmid with ligated insulin gene is inserted into the bacterial cell. Bacteria that multiplies in the culture medium produce inactive insulin. Active insulin is produced from this. Question number 28. A. Name of this technology. Answer. Genetic engineering. Question 28B. Basis of this technology. Answer. Genes can be cut and joined. Question 28C. Genetic scissors, genetic glue, vectors. Answer. The enzymes used to cut genes are known as genetic scissors. Example, restriction endonuclease. The enzyme used for joining is called genetic glue. Example, the enzyme ligase. A gene from one cell is transferred to another cell by using vectors. Vectors contain the ligated genes and they enter into the target cells. Examples of vectors: plasmids in bacteria. Question 28D. Scope of this technology. Answer. Gene therapy, genetically modified animals and crops, forensic test. Question 28E. Demerits of this technology. Answer. The genetically modified varieties cause harm to indigenous varieties and may cause health issues to humans. So the first point is genetically modified varieties are threat to indigenous varieties. Second point, genetic modification is a violation of rights. Genetic modification is an intrusion upon the freedom of living beings and it is a violation of rights. Third point, genetically modified pathogens are a new challenge and it is used by enemies and thus that type of wars are called bio wars, and in bio wars These genetically modified pathogens are used as bioweapons, and this is a threat to the existence of human beings.